Welcome back to part 2. This week, it's only chapter 6, so this should be the last video that you need this week. Again, don't watch this before the exam. Wait till after exam 1. Watch it before exam 2. That would be a lot. So we said plasmids in the last video. It was that ring of extranucleoid DNA. It came from somewhere else. It's a little ring of DNA inside of a cell. Maybe it's gone through conjugation, but it's not a high-frequency recombiner. So it's got this loose piece of DNA hanging out in its cytosol. Bacteria are simple. It doesn't need to have its DNA in a special place to use it. For us, our DNA is contained in our nucleus, and all of the proteins and enzymes that copy and use and transcribe that DNA is contained in our nucleus. But for bacteria, all of those proteins are just like loose, hanging out. So if there is DNA, and that DNA has a promoter, a starting region, that DNA will be used. It will be transcribed, turned into an mRNA transcript, and it will be translated into something that the bacteria can use or kills it one way or the other. When we talk about plasmids, it's sort of outside of the chromosome. I like to say extranucleoid because it's really, I don't know, it makes me think more about the nucleoid, but it's not in the bacteria's chromosome. And we classify plasmids by what code is on them. I have 2 kinds of plasmids to talk to you about. I'm going to have R plasmids and another one a little bit later. R plasmids have 2 parts to them in their DNA structure. They have an RTF. This is the resistance transfer factor. The resistance transfer factor is the gene information for causing the plasmid to get transferred. So it can be passed on. The other part is called the R determinant. This is the gene that is expressed in the bacteria. Often this is something like antibiotic resistance or a defense against heavy metals. There's other things, but these 2 are the most important for us as humans. Why is it important or actually a problem if the plasmid in a bacteria has antibiotic resistance? Well, think back to conjugation and think back to the idea of horizontal gene transfer in general. When I have horizontal gene transfer in bacteria, they are passing this plasmid from one individual to the next individual, even though they did not inherit that DNA. If that R determinant region has the code to be antibiotic resistant, to not die when I use an antibiotic, and it can be transferred to other bacteria in my lab or in my body, that's a problem. Bacteria can acquire resistance to metals or antibiotics from their peers. They do not have to spontaneously mutate to be antibiotic resistance, which has allowed antibiotic resistance in bacteria to spread among various populations and various species way more quickly than we would have anticipated, understanding just the idea of spontaneous mutation or adaptation. What would happen if I have a plasmid and it just has the RTF but no R determinant? And if you're still thinking, pause me because I'm going to say the answer. It wouldn't confer any resistance or anything favorable. It would just transfer itself. So if it's just an RTF, this is just a plasmid that's transferring itself from bacteria by bacteria. I think it would be energetically costly for the bacteria that received it and that spent it. Over time, I actually think that natural selection would take that out because this bacteria is spending time copying and transferring something without any benefit at all, so that it's losing the energy and gaining nothing. Whereas the R determinant means that it's using energy to copy that, but it's also gaining something in the end of that story. The COL, cold plasmid, is a little different than the R plasmid. It encodes for proteins that are toxic to bacteria that don't have the same protein or plasmids. It actually releases a compound, a protein called colicins. They kill the bacteria if they don't have this plasmid already. We are going to use this as an artificial vector. A vector means something that carries something from place to place. Mosquitoes are vectors for malaria. Mosquitoes don't really get malaria, they just transfer it from place to place. We use these plasmids as a vector for cloning. We transfer them from one cell to another cell, carrying the cloned information that we want, and they also have a code to make sure that they wipe out anything that is not them. I have two more examples of horizontal gene transfer where something, either a plasmid or just DNA, is transferred from one individual bacteria to another. Transformation is the process of taking just loose DNA from the environment, and bacteria will pick it up. And when they pick it up, if it is a full gene with a promoter on it, they will express it. You saw this in Bio 1 in Griffith's experiment. So I put, this is the slide I used for Griffith's experiment in my Bio 1 class. But Griffith was trying to understand how DNA, if actually DNA was coding for anything at all. He was working in about 1928, and at the time, no one knew what carried the code in the chromosome. They knew that the chromosome exists. Morgan and Mendel and Studevart and all those people had been doing their research for a very long time, but the actual what is on the DNA that is carrying the code had not been answered. Or if it really even worked in animals. Griffith actually did an experiment with two different strains, so two different varieties of this is a Streptococcus, so it's a kind of bacteria that kills mice. But there's two versions. There's the kind that doesn't kill mice and the kind that kills mice. The difference between them is the not killing one is rough from a biologist in 2026 or in the future view of that. That means that this one does not have a protein coat protecting it on the outside. The smooth strain kills mice. It actually has a coat on the outside that inhibits the ability of the mouse's immune system to attack it. Griffith didn't know that, but we know that, so it makes it make more sense. When Griffith injects the rough strain bacteria into the mouse lives, right? This is not mouse-killing bacteria that doesn't kill mice. This is not surprising. When Griffith injects this smooth strain that kills mice into a mouse, the mouse dies. Also not a surprise. If you put mouse-killing bacteria in a mouse, it dies. When Griffith denatured the bacteria, he killed it in heat, and he destroyed this outer coat. And he injected the killed smooth bacteria into the mouse. Well, it turns out dead bacteria don't kill mice. Also, not a surprise. What was a big surprise is when Griffith took the rough strain, so the not mouse-killing bacteria, and he mixed it with the smooth strain, the other not mouse-killing bacteria, and he injected into a mouse. The mouse died. Which is a surprise. Because if you take two kinds of not mouse killers and put them together, they definitely should not kill the mouse. What Griffith said was happening was that the dead smooth transformed the live rough into mouse killers. Out of control. What we now know happens here, so just changing the picture here, is that the bacteria in general, many of them have a protein on the outside of them. You see this receptor site here. Have a protein on the outside that allows them to identify exogenous DNA, or this is DNA from the surrounding. It's just DNA that's floating around in the soup. When it identifies this, it will initiate pulling the DNA that it just found into the bacterial cytosol. If that is an entire gene, as long as it has a promoter, the bacteria will then access it. It'll start using it. In Griffith's experiment, what happened is he popped the smooth bacteria and their DNA fell out. The mice not killing rough bacteria, picked that up with its receptor site, drew it into its chromosome, and incorporated that piece of new DNA that it picked up in the outside into its original genome, and then expressed it. This cell, when it has been transformed, has a new piece of gene in it. Now, I want to explain this a little bit beyond what you did in Bio 1, because I think you did that concept in Bio 1, maybe not the word exogenous, which is why I made it red. Not all bacteria in the population will do this. This is actually pretty rare. We need them to be competent. One of the challenges we're going to have as we go through our pGlow experiment is we're not going to use conjugation. We're going to use something closer to transformation. We're going to expect our bacteria to be competent, or at least hope that some of them are. They're able to pick up that DNA from the surrounding environment. When they do it, they can then express it. When the DNA enters into, I think I have it on the next slide, yeah. When the DNA enters in through this receptor and we bring it in, it is DNA, so it has two different strands that are running anti-parallel, and they are bounded across the center with their A's tied to their T's, hydrogen bonds, and their G's, ignore that. Their G's and their C's, and their A's and their T's. Hydrogen bonds holding the two strands running in anti-parallel directions. When they enter the cell that it has about to be transformed, those two sides of the DNA separate. And so each side remember, they run anti-parallel to each other. And we're going to cover this again in case this is not familiar. They run in opposite directions and they form base pair matches with hydrogen bonds. They separate away from each other and one side randomly degrades, meaning it breaks down. The other side finds something that is really, really close, not identical, but really, really close in sequence to it on the genome of the bacteria itself, and it binds to it. At this point, this recombination here is called, this region becomes a heteroduplex. The one side of the original native, bacterial DNA degrades away, and this transforming strand bonds in and fills this other side. As this cell then lives its life, it's got, bear with me here, I'm going to draw this out. It's got one side that it just brought in. We're going to make that T-A-G. And we're going to change my ink color to orange. The original bacteria's DNA is kind of bound to it. Let's make this an A, a C, and a C. It's not exactly right, but it's close enough to be stable. This region here is now a little bit different from its source and code of DNA. This is a heteroduplex. The next time this cell goes through binary fission, it separates the strands of DNA. It makes copies of those DNA strands. It goes through DNA replication. One of those copies will be of the inserted DNA. One of those copies will be of the not inserted DNA. And so half of this bacteria's babies will possess the new gene that they brought in through their receptor site. And then they'll go through binary fission. I'll end up with 1 billion of them that are transformed. Sometimes genes get linked together in this case, and more than one gene is brought in. Depends on what happens prior to transformation to the bacteria that is the donor bacteria. So a little practice with me problem in the middle. I know it's in the middle of the lecture. There's actually more after this, but a little bit of practice with me. In the transformation experiment, we have donor DNA. This is what was picked up. This is the exogenous DNA that was picked up from outside. That came from a prototroph, something that is able to digest everything properly. It can digest, it has the alleles for digesting A, it has the alleles for digesting B, and it has the alleles for digesting C. The recipient, the one that picked up the gene, was an auxotroph. An auxotroph means that it is missing some of the genes it needs for nutritional completeness. It means I have to feed it from a medium if I want it to survive, where I provide those nutrients for it. So for this thing to live, I need to provide A, B, and C. For the prototroph, I don't need to provide anything. It'll grow at a minimal medium. So you don't have to tell me how many. You don't need to tell me what is happening here. But I just want a general idea of what these kind of babies mean to you. Donor was A plus B plus C plus, and the recipient was a triple auxotroph, A minus B minus C minus. You're not getting a number, just a general gut feeling like what's happening. This is the highest number out there. Higher frequencies suggest closer linkage. Here is the donor type. This is what came from the one that gave the DNA in the solution. Its frequency is very, very rare. This actually suggests that A, B, and C are not really closely linked to each other. A and B, A and B to me look like they are closely linked. A and B positive stay together at really high rates, but C seems to be much further away on the chromosome. A and B negatives stay together at really high rates. So a little bit of segue to the virus itself. Bacteriophages are viruses that infect bacteria. They really look like this. They look like alien ships. They're primarily made out of DNA here and protein. The protein coat with some DNA inside of them. Bacteriophages, they move from bacteria to bacteria. They inject their DNA into the bacteria, and then they also have some substances that they inject into the bacteria that break up the bacterial genome and make it so only the viral genome still exists. This is the T4 bacteriophage. It is also one that affects E. coli. It has about 150 genes that it carries for the capsid, but also for other things it needs to do to take over the bacterial mechanisms to making proteins and copying their DNA. The replication cycle of the T4 phage starts when the phage binds to the bacteria. If you took micro, you did this. This is nothing new. I'm not changing the story at all, but it's not required for this class. So the T4 bacteriophage is what's called virulent. It infects the E. coli, it undergoes the lytic cycle, and it kills the E. coli and makes new phages. It starts when the phage, so here's the phage right there. He's super cute. For a virus. He attaches with protein to protein contact to this bacterium. The phage injects its viral DNA into the chromosome, into the bacterial cytosol, and some enzymes that actually degrade the host chromosome. So this thing gets degraded. While the phage's instructions are inside the bacteria doesn't know that it was phage. All it knows is that it's DNA. It doesn't know anything. It's just chemical reactions. The bacteria starts reading the code on the viral DNA and executing it. It starts expressing it. So it goes to transcription, translation. It makes phages. It follows the code in the DNA that it's just been given, and it takes those phages and it assembles them around the DNA copies of the phage viral DNA. When it's done, there's about 200 new phages packed inside of a completed cycle. The bacteria is so full that it actually pops. This is called lysing, Lyse. It equals pop. and all of the new T4 viruses pour out and are ready to infect the next stage. The lytic cycle starts with attachment and ends with lysing. We can look at this in a culture in the lab by doing something called plaque assays. Phages are really tiny. Bacteria are really tiny. We can't see this, but we can make serial dilutions of a bacteriophage culture and then see how it works on a plate of bacteria. So here I make a serial dilution, which means I start with a lot, and each time I increase the amount of water or solution per bacteriophage culture and a measurable amount in each dilution. So here I have 1 milliliter of phage, the concentration at 1 milliliter for phages, and then I dilute them and I dilute them and I dilute them. I add 1/10 of a milliliter each time until I've diluted it enough to be usable for us. I have here a lawn of bacteria. So a little bit, the color in the pictures of the lab book look the same to me. This is all bacteria growing on this plate. It's completely covered in bacteria. And I expose them to different levels of bacteriophages. The 10 to the 3 here has so many bacteriophages that it killed all of the bacteria. I can't understand that. I can't read that as a number. The 10 to the 7 dilution has all bacteria and none of them have been attacked that I can see from the phages. I can't read either of these. They don't give me any information. This one is useful. When I put this dilution of bacteriophages on my lawn of bacteria, the bacteriophages are kind of spread out and they start attacking the bacteria and reproducing. These little gray areas represent places where the bacteria has been killed. I can read each plaques or places where the bacteria has been killed. I'm writing it out. Each one of those I count as a plaque. I can read and calculate the concentration the same way we did in the last lecture when we were concentrating bacteria and looking for colonies. I can look at my plaque number per milliliter times my dilution factor of my measurable plaque, so 10 to the negative 5 times 23, and that will give me the initial density of phages in my solution. I want to point out here, this is really counterintuitive because of the way we do it one way and then the other. In the first one, where we were counting bacterial colonies, the dots were bacterial colonies. In this one, in the pictures, the dots are plaques. The dots are where there are no bacteria. These are more like killed spots on the lawn. I don't know why that happened. The T1 bacteriophage, not T4, which is what we were just looking at, T1, the one that infected our colonies in the very beginning of chapter 6, it's called a temperate phage. Temperate means that it can temper its response depending on the health of the bacteria that it's infecting. It can undergo the lytic cycle. It can infect, explode the bacteria at the end. Or it can undergo the lysogenic cycle. The lysogenic cycle is sort of like being a wolf in sheep's clothing. It's hiding. It's going to incorporate itself into the bacteria so that it gets passed on to the bacteria's babies. And now those babies have the gene for expressing the phage, but it's not turned on. So because it gets asked, and it's not covered in your textbook, it's covered in other classes that I teach though, so I'll give you the hints on it. What happens for real is when the phages that are temperate are exposed to a bacterial colony, if that bacterial colony is stable and healthy, it's using a lot of glucose. And when it's using a lot of glucose, that means in its cytoplasm, it actually does not have a lot of a molecule called cyclical AMP. cyclical AMP results from using up all of your ATP. If that bacteria is stable and eating well and everything is going well, the temperate phage will enter the lysogenic cycle. It knows, quote, quote, knows, that that bacteria is likely to go through binary fission. And so its best strategy evolutionarily is to hide inside that bacterial's genome. It binds to the outside of the E. coli host. The arrow is going this way. This process of binding here is called adsorption. It binds and locks together. The locking together triggers the lysogenic cycle in this super happy bacteria. The temperate phage injects its DNA, and that DNA hangs out inside of the bacteria's chromosome. It can, here, incorporate into the bacterial chromosome, and it becomes a latent gene. Latent means it's hiding. As it goes through its life cycle, it can be passed on into this bacteria's babies. These are baby bacteria. And each one of them will inherit a chromosome from its parent that incorporates the bacterial chromosome and this waiting and hiding gene for the virus. When the viral DNA is ready to pop out, the bacteria is stressed. It's running out of food. It's not likely that it's going to go through binary fission. The lysogenic cycle will trigger into the lytic cycle. The lytic cycle is when the bacterium is actually expressing the phage DNA and will make baby phages. This is a bit counterintuitive. The lysogenic state never makes phages. There could be theoretically a situation where a virus enters into the lysogenic cycle, the lysogenic state, and never emerges. I would never even know it was there because it would never make phages. This is just about hiding. Hopefully this will work. This is just an animation, but I want you to watch both sides of this. I'll let it run 2 or 3 times and then you can rerun it again, and I'll link I'll put this in so it's a slide that plays when you guys have the slides. The lytic and the lysogenic cycle, these little colorful guys are the cells. The lytic cycle makes little phages and explodes out of the bacteria. The lysogenic cycle, it is still passing its genes on, but it's taking advantage of binary fission. Initially, it seems like in the lysogenic cycle that there are fewer opportunities to pass your genes on. But ultimately, each one of these poor, unfortunate cells will enter this little incorporated thing is going to be activated. It'll remove itself from the genome of the bacteria and it will enter into the lytic cycle. That little thing, this virus DNA that is incorporating right there, is called a prophage. It's a hiding viral genome. Transduction is the process of this being hijacked in order to cause bacterial recombination. When I have this, so third story about viruses infecting bacteria, when I have a phage that attacks a bacterium, the phage injects its DNA and then it runs the lytic cycle in this example. The DNA of the host is destroyed. The virus DNA is used to assemble and build new baby viruses. Those baby viruses they're supposed to have viral DNA in their capsids inside their heads. But some of them, because there's no guy in here organizing everything, some of them, like this guy, actually incorporates a little bit of the bacterial genes instead of viral genes inside of its capsid, its head. When this guy releases and he goes off to complete the replication cycle the next time, it releases into the solution. It still goes through binding where it binds to the next, I don't want to say victim, but the next bacteria that's going to be infected. It injects the DNA inside of its head, but this DNA is bacterial DNA. It also has all of the proteins for splicing and dicing. And what will happen is sometimes this bacterial DNA that came from over here is inserted into the recipient. Now it just acquired a new gene. It does not get baby viruses because it never got the code for baby viruses. What it got was the code for something this bacteria had and this initial bacteria did not. The phage attacks the bacterium and undergoes the lytic cycle, but when the phage is released, some of them contain bacterial genes, and that allows for genetic diversity. This is called transduction. I had a student one year tell me the way she remembered this was it sounds like abduction. And this kind of looks like an alien ship that comes along and like steals something and it takes it to another place. It's kind of like it's being abducted by aliens. It's being transducted. A little tiny, I'm actually gonna wrap it up with this last idea. You have heard about these stories before if you took Bio 1, if you did any sort of like back in your memory of where the understanding of what DNA actually was. Some of these things that we've just talked about played a role in how we actually identified DNA as the molecule of information. If you remember back, and if not, I think we covered again in a couple lectures, Hershey and Chase's famous experiment where they identified DNA as the molecule of information. Margaret Chase she tagged different types of bacteriophages with different types of radioactivity. So she labeled the DNA in some cases, and she labeled the protein in other cases. When she labeled the protein with radioactivity, she could trace it through the lytic cycle and see what the baby phages here, the little new phages, came out with. If she labeled the protein on the outside of the phage, those new phages were not radioactive. If she labeled the DNA with radioactivity and allowed the lytic cycle to progress, the new phages, the babies, would come out as radioactive. Her experiment really illustrated the fact that DNA was the code that was building these little guys. DNA was actually conveying a code. So as we go forward, keep that in the back of your head. I did not do the whole chapter here. Some of this is outside the scope actually of our class, getting into chapter 6. You only have chapter 6 this week. I hope it's not too stressful. Practice for the exam. I posted some practice things for you to work on. We'll review in the Tuesday lecture if you haven't already had that. And then the exam is the only thing I expect on the rest of the week. And then we'll start doing some actual lab experiments where we'll look at this in real life, some of these things, as we start into the week after that. I hope you're having a good time. Please ask me if there's any questions, and I'll see you in real life or in the next video. All right, bye-bye.